Good morning. Let's discuss on chemistry of amino acid. As we all know, amino acids are building blocks of protein, and proteins are of paramount importance for biological system. All major structural and functional aspects of body are carried out by protein molecules. Very, very important among all the macromolecules of the living beings. All proteins are, you all know, they are the polymers of amino acids, and around 300 amino acids occur in nature and only 20 of them are seen in the body, and most of the amino acids are alpha amino acids, which I'll tell you what are alpha and beta, different vocabulary associated with chemistry of amino acid we'll discuss here. So let's continue. Today's learning objective are: classify the amino acid based on its structure, nutritional requirement, and metabolic fate. We'll come to know about the isoelectric point, and we'll be enumerating the reactions due to different functional groups of amino acids. Major macromolecules in the body are carbohydrate, protein, and nucleic acid, as we all know. And this is the general structure of amino acid. The 20 amino acids found in proteins have a carboxylic group and an amino acids group bound to the same carbon atom. So you can see the structure here. This is the structure, amino group, carboxylic group. This is the carbon, which is alpha carbon. The carbon which is directly attached to the carboxylic group or the functional group. There are two functional groups. This is the higher priority functional group. The carbon is attached directly to this. So this carbon, this is alpha carbon, and this is the side chain. So this is the general structure of all the protein. Uh, Carbon, hydrogen, and oxygen. Nitrogen are the basic constituent elements of the amino acid, and out of this, 16% is alone is nitrogen. Amino acids differ from each other in their side chain or the R groups attached to the alpha carbon atom. So the 20 amino acids of protein are often referred to as the standard or primary or normal amino acids. One of the 20 amino acid, the exception is proline, where instead of like it is amino acid, we call it amino acid because the amino group is in the secondary amino group form. And we'll see, discuss later on, we'll see the structure of this. This is the amino group, and the amino acid not in the free form, but it is a part of this ring, proline ring, 5 carbon ring, pyrrolidine ring. This ring is known as pyrrolidine ring. So this is the constituent. It's not in the free form as in case of other amino acids, while carboxylic acid group is free here. All naturally occurring amino acids are optically active except glycine. No asymmetric carbon is present in this molecule, which is optically inactive. You can see the structure of amino acid. This is the structure of glycine. Glycine has two hydrogen. Two valencies are common, so it is optically inactive. While in rest of the amino acid, whether it is D or L, all four valencies are different. That's why this is these are optically active. The two free D amino acids, D-serine and D-aspartic acids, have been found to be present in brain tissue. D-alanine and D-glutamates are found in some bacterial cell wall. Rest of the amino acid found in the living being, especially in ours, it is your L amino acid. Classification of amino acid has been done based on the structure, based on side chain, based on metabolic fate, and based on nutritional requirement. So we'll see one by one. Amino acid classification based on its structure. So we have classified accordingly. So aliphatic amino acids, first classification, sub-classification of this group is aliphatic. Then we will come on to the aromatic, heterocyclic amino acid, amino acids, and derived amino acid. So this is the blueprint of the structural classification of amino acid. Aliphatic amino acids are further divided into three groups: monocarboxylic acid. In this category, only neutral amino acids come. Then monoamino dicarboxylic acid, because the predominance of acid group, these are acidic. Here the third is diamino monocarboxylic acid. Here the predominance of amino group, diamino group. One is present in the side chain and one is a basic part of basic skeleton of all amino acid. So this is basic. Amino acid classification में first classification we will see the aliphatic amino acid. So monocarboxylic acid. All the members of this group are basically neutral. So you can see the structure of simple amino acids. COH, and this R is H. So if R is H, it is glycine. So two H are there, so it is optically inactive. I discussed. And here the R group is alanine, CH3. So that's why its name is alanine. So both are neutral. Then valine, leucine, and isoleucine are branch chain amino acid. So in the branch, this R group is in the form of branch, side chain branch. So this is isopropyl group in valine. Then this is a tertiary butyl group attached here. This is also a normal butyl group attached here. It's secondary, through the secondary valency it is attached. This is isoleucine, valine, leucine, and isoleucine. Then second is monoamino monocarboxylic acid. We will discuss on hydroxy amino acid. Hydroxy means the side chain contains the hydroxy group. Basic skeleton remains the same. You can see in the box, pink box, it's given. Only change is there in R group. R is here. If you see the structure of alanine is having a methyl group here, you replace one of the hydrogen by OH group, so it becomes serine. So very easy to memorize the structure. This is the structure of serine, and then in threonine, one more hydrogen from this you replace by methyl group. So this becomes threonine. So that's how it has two. What change happened here? You can notice here the two chiral carbons. Instead of one is here only. But in case of threonine, there are two chiral carbons. One is here in the side chain. Then sulfur containing amino acids. So in the side chain, there is a sulfur group present. So CH2SH is the R group here. And this is cysteine, the molecule cysteine. One more CH2 when you add on, it becomes the and a thioether linkage is there, very prominent. And through thioether linkage, there is a attachment of methyl group. It becomes methionine. So keep remember the structure of this methionine. Amino acid with amide side chain. So in the side chain, there is amide group. So the two, this is the amide group. One is asparagine, only one carbon chain is there. Here the two carbon chain and then amide group. This is glutamine. Keep remember. All these are neutral amino acid basically. Now move on to the acidic amino acid, because they have predominance of carboxylic acid group. So you can see here in the structure, the basic structure is this in the box, and the R group is CH2COO. So that means in the side chain also there is additional carboxylic group is there. Here also there is a alpha carbon, then beta carbon and gamma carbon. So gamma carboxylic acid group is present in the glutamic acid, while in the aspartic acid it is the beta carboxylic acid. Keep remember. So then diamino monocarboxylic acid. These are the basic amino acid. And arginine, the representatives are arginine and lysine. Arginine, you can see it here. This is the basic skeleton of all amino acids. C alpha carbon, COH group, and the NH3 group. In the side chain, there is a CH3, CH3, CH3. Like three carbon chain is there. And here at the end, there is a guanidium group, highly basic group because of presence of three amine groups, right? So in the case of lysine, And this side chain is having the 3 carbon chain and the only amine group is there. Okay. So arginine and lysine. Arginine has the highest grade of basicity among all the amino acid because of presence of this guanidinium group. Aromatic amino acids, they are having the no, as the name suggests, aromatic. So the aromatic ring is present in the scaffold. You can see it here. This is the basic structure, and R is CH2, and this is the benzene ring. So this is the phenylalanine. Matlab, if you know the structure of alanine, alanine is having CH3 group here. From the CH3, you replace one of the hydrogen by this benzene ring. So it becomes phenylalanine. Then if you add on one hydroxy group in the side chain at the para position, it becomes tyrosine. This is the structure of tyrosine. Then heterocyclic amino acids. In the heterocyclic amino acid, in the side chain, some heterocyclic ring is present. So these are all can be called as derivative of alanine. Hydrogen you can replace, and one of the hydrogen is replaced by this bulky group. This is the indole ring, one benzene ring, and one 5 carbon ring. 5 carbon ring. Both are attached, and via CH2 group, they are linked to the basic skeleton of amino acid. This is the structure of tryptophan, heaviest amino acid among the all. Then another member of heterocyclic category is, and this is your histidine. Histidine, it's having in the side chain there is a imidazole ring through nitrogen containing. This is also a heterocyclic. This is also a aromatic ring, kind of aromatic ring, because this sextet is complete. Aromatic sextet is complete. So because of localization of these four electron of the pi ring and the two electrons of the lone pair. So all them total 6 electron in the movement conjugation. So this is imidazole ring. Via CH2 it is linked. This is the structure of histidine. Now move on to the exceptional amino acid, that is amino acid. So proline is the only amino acid which doesn't have the amino group as a free, but it is a part of 5-membered ring. This ring is known as pyrrolidine ring. Rest of this is the alpha carbon, and this is not alpha carbon. This is COH, and this is the alpha carbon. Okay. Now move on to the derived amino acid. They can be of 3 types. One, derived amino acid which are present in protein. Second is that they are not present in protein, they are independently existing and performing some functions. Then non-alpha amino acid. So let's see one by one. Derived amino acid, the one who found in proteins, they are hydroxyproline, hydroxylysine, very, very important derived amino acid, which are important for cross-linking of collagen. Their absence lead to abnormal kind of collagen. Then gamma carboxy glutamic acid important for clotting. Then methylated and acylated proteins. They're important for ribosomal protein and histone formation. Not the protein, the amino acid not found in protein. So these are ornithine independently existing there. Ornithine and citrulline, important intermediate of urea cycle. Then homocysteine, it is a sulfur containing amino acid, very, very clinically very important in cardiac disorder. Then non-alpha amino acid, it is first member is gamma amino butyric acid derived from glutamic acid. If you remove the amine group, so it becomes a gamma amino butyric acid. Only one amine is lost. Another amine which is present in the side chain, it remains intact. So it becomes the gamma amino butyric acid. Beta alanine, it is important in a component of coenzyme A, while this GABA is an inhibitory neurotransmitters. So this is in nutshell the slide summarizing all the amino acid based on the structural, based on their structure. This is the compilation of all derived amino acid. GABA and dopamine are neurotransmitter. Histamine is important in allergy. And then thyroxine is a hormone. Cycloserine is a drug. Azaserine is a drug. Uh, Ornithine is important intermediate of urea cycle. Citrulline is also an important intermediate of urea cycle. These are important anti-cancer drugs. Spatial groups in amino acids. So you have seen arginine is having guanidinium group because of its the highest degree of basicity is present. Then phenylalanine is having the benzene group present in the side chain. Tyrosine having the phenol group present in the side chain. The tryptophan is having the indole ring in the side chain. Histidine is having the imidazole ring in the side chain. So they can uh, there is a special attention on these groups present in the side chain, and because of them, the amino acids show some different or some very special kind of characteristics. So this is the classification based on polarity. Amino acids have been classified into the hydrophilic, that is polar, and non-polar groups, non-polar categories. In the polar categories, they can be negatively charged, they can be positively charged, they can be uncharged. So negatively charged, aspartic acid and glutamic acid. If you remember the structure in the aspartic and glutamic acid, in the side chain there is a carboxylic group, and this carboxylic group, once it loses its hydrogen, it becomes negatively charged. Similarly, in the positively charged category, lysine, arginine, and histidine come. So in the side chain, there is a amine group present, and this amine, when like it's being hydrogenated or it's being protonated, it becomes positively charged. Uncharged category mein your serine is there, threonine, asparagine, glutamine, cysteine, and tyrosine. So these are all neutral group present in the side chain. In serine, there is a thioether linkage, methyl thioether group is present. In threonine, there is an hydroxy group present. Then asparagine is having the amide, glutamine is having the amide group, cysteine is having the SH thiol group, tyrosine is having the hydroxy group in the ring. Then hydrophobic category mein non-polar, aliphatic side chain and aromatic side chain. So in the aliphatic side chain containing amino acid are glycine, alanine, valine, leucine, isoleucine, proline, and methionine. Then aromatic side chain is having the phenylalanine, tryptophan. So classification based on side chain characteristics. Acidic, they can be acidic or basic or the neutral. So acidic may there is, I have already told you, they contain extra carboxylic group in the side chain. So they can be asparagine, then you have a glutamine, and third is tyrosine is not very acidic because of some other factors like conjugation and because of presence of benzene ring, it may show slight acidity. Then basic is arginine, lysine, and histidine. Neutral, rest are neutral. Then classification based on metabolic fate means the classification has been done on the basis of their metabolic end product. What product did they either produce the glucogenic precursor or they do produce the ketogenic precursors. So on that basis, the amino acids have been classified. So you can see it here. The amino acids being glucogenic or ketogenic based on the type of intermediate that are formed during the breakdown or catabolism. Catabolism of glucogenic amino acid produces either pyruvate or one of the intermediate of the Krebs cycle. Catabolism of ketogenic amino acid produces the acetyl coenzyme A or acetoacetyl coenzyme A. All these are the precursor for synthesis of fats or ketone bodies. So you can see it here. For example, the leucine, isoleucine, and tryptophan. The leucine is predominant or exclusively it is your ketogenic. It is the only amino acid we can say it is a ketogenic. Rest are isoleucine, tryptophan, both they are both ketogenic as well as glucogenic. So they are synthesizing here acetyl coenzyme A, leucine, lysine, phenylalanine, tryptophan, tyrosine. They are all synthesizing your acetoacetate. The alanine, cysteine, glycine, serine, threonine, and tryptophan. They are ending up to the formation of pyruvate. Aspartate, phenylalanine, tyrosine, their metabolic intermediates can form the fumarate, isoleucine, methionine, threonine. So it is forming the succinyl acid. So similarly, arginine, glutamine, glutamate, histidine, and proline. So these are alpha ketoglutarate. They are forming the alpha ketoglutarate. So based on their metabolic weight, they can be of three types. One is ketogenic, one is glucogenic, and third is they are both ketogenic as well as glucogenic. I can give you one example. Isoleucine is here also. It is synthesizing the succinyl acid, which can end up to oxaloacetate, and then malate is there, and malate to oxaloacetate in cytoplasm, and further it reverts back to the reverse step of glycolysis, and it is ending up to the formation of glucose via gluconeogenesis. So it is purely glucose, it is glucogenic here, isoleucine, but here at the same time this isoleucine is synthesizing acetyl coenzyme. So this is, it shows it is ketogenic as well. So it is both ketogenic as well as glucogenic. Then based on the classification, based on nutritional requirement, so they can be of two type, essential or non-essential. Essentials means they cannot be synthesized in the body. So if they are not available in the food, so we take them from the food. We supplement them in the food. Why they are important? They are very important for growth and development, because every cell membrane, every membranous structure is just consist of proteins and lipids. So these proteins, the complete proteins, are very much required for growth and development. If any of the single essential amino acid is absent, so proper growth will not take place. Then partially essential category, there are arginine and histidine. Only two amino acids are non-essential. Rest are non-essential amino acids, which we can synthesize in our body. So ILTL, MPTV, isoleucine, leucine, lysine, you can see it here. Essential में isoleucine, leucine, threonine, lysine, methionine, phenylalanine, tryptophan, valine. You can make some mnemonic. You can easily remember it. Partly essential category में arginine, histidine, and rest are your non-essential, because we can synthesize in our body. Then properties of amino acid. They have taste. So this glycine, alanine, valine, serine, tryptophan, histidine, proline are sweet in taste. Leucine is tasteless, while isoleucine and arginine are bitter. They have higher melting point because being polar molecules, because of polarity, high degree of polarity is present. The amino group, one is amino, one is carboxylate, two antagonistic groups are present. So it is highly polar molecule, and that's why the melting and boiling points are very high. A melting point, they are solid, so their melting points are very high. Water soluble and lipid insoluble. Now move on to the isoelectric point. It exists as zwitterion, neither cation nor anion. I will show you how. The pH, isoelectric pH, at the pH at which there is no net charge is known as isoelectric pH. The positive and negative charges cancel each other. No mobility in the electric field, and solubility and buffering capacity are minimum. So these are few important feature of the amino acid at the isoelectric pH, and we can have very so many advantages because of these characteristic at isoelectric pH. We can easily separate the different categories of amino acids and protein based on their isoelectric pH. You can see it here. This is the amino acid. When the amino group is positively charged at one time, so at low pH, definitely at low pH, means highly acidic condition. So amine group is protonated. Second is in the second stream, this is the carboxylic group, which is in the anionic form. So definitely it will be at the high pH because some basic condition will remove this hydrogen group proton from this carboxylic acid. It is negatively charged. So this is the zwitterionic state where it can we can call at like intramolecular exchange of shifting of protons right from this carboxylic group to amine group. So it becomes negatively charged, it becomes positively charged. And because of presence of these two extreme positive and negative groups, the total charge on this particular molecule is nil, cancelling each other. So this state is known as zwitterion. Instead of zwitter means joined with each other. And so this is the exceptional case where both the ions are joined. They can never be separated. You cannot separate them. That's why its name is zwitterion. And zwitterionic form is always present in the isoelectric pH range. So what is the isoelectric point? Try to understand. What is the isoelectric pH? I have already told you in the last slide, like the pH at which there is no electrical charge onto the amino acid because an intramolecular transfer of hydrogen has taken place. So negative charge is present in the form of carboxylate ions, and the positively charged amino group is present in the form of positively charged amino group. So there is no net charge, no mobility in the amino acid in electric fields. Solubility भी minimum होती है, then precipitation is maximum. So taking advantage of all these characteristics, we can exploit this isoelectric point or pH for separation of various amino acids as well as the protein from their mixtures, which becomes very difficult to separate by other technique. Then buffering capacity is minimum at PI, maximum at PK1 and PK2 because they are in one of the ionized forms. Either they are positively or negatively charged. So pK of imidazole is 6.1, which is very close to the pH of blood. That's why they are very good buffers. Imidazole albumin contains the very good amount of these imidazole, and because of these, the albumin contributes a lot to the buffering activity of the blood. They maintain the pH of blood. So buffering action of amino acids and proteins. So minimum buffering capacity at PI, the maximum buffering capacity in around PK1 and PK2. The PK value of imidazole group is histidine is 6.1. Keep remember, effective. It is a very effective buffer at physiological pH. Albumin contain the 16 histidine residue, which is a very significant amount, and hemoglobin contain 38 histidine residue. And because of presence of very good proportion of histidine, albumin and hemoglobins are both very good pH regulator of the blood. They have good buffering capacity. You can see it here. The pH, this is the titration curve showing titration curve of glycine. So you can see it here at the low pH. The titration is being performed via NaOH. So red cell formation titration is being performed. But there is one difficulty. We all know that amino acid cannot be exactly titrated. If, for example, the 1 ml of NaOH, 1 N glycine, 1 normal glycine is titrated against 1 normal of sodium hydroxide. So the alkali requirement will be less than 1 ml. Why? Because the hydrogen ion released by ionization of carboxylic group are partly taken up by the amino group. So internal neutralization happens. So to circumvent this problem, in Sorensen's what he did, he used formaldehyde, which converts formaldehyde. What it will do? Formaldehyde will partially protect. It will temporarily protect this amino group, in the form of dimethylol derivative. So we can have a complete titration against the Cu carboxylic acid group. So you can see here, this is the pH range, this is the degree of titration. So as we are raising the pH right from 0 to 13, 14, we can see it, titration is being performed by the dilute solution of, we keep on changing the pH of NaOH. You can see right from 0 we started with, so from 0 to around 2.4, there is a, it remains in this particular form. The glycine remains in this form where the amine group is protonated. As we are increasing the basicity, slowly raising the pH to 6.1. At 6.1, there is a complete ionization, both carboxylic group and amino group in the ionized form. This is negatively, this is positively. Further, we are raising the basicity right from adding, we keep on adding the NaOH slowly, one normal solution. So around 9.8, another like another this COH remains in the negative form, but one more proton is removed from this amino group. So this is the titration curve, and pK1 and pK2 are the two ranges where there is a maximum buffering capacity. Optical activity. All the amino acids are optically active except for glycine, as I have already told you. And because they all have chiral carbon atom, one carbon, alpha carbon is having the valencies, four different valencies attached here with. So this, and beside this, capital and capital D designation is based on, it is an ambiguous system of naming to confer the absolute configuration to the amino acid L and D. So this is based on the like correlating the structure of the conformational structure of this stereochemical structure of amino acid with the L-glyceraldehyde and the D-glyceraldehyde. So it confers the L and D absolute configuration. L are very common. Maximum amino acids are, all 20 amino acids are L amino acid, while D are very unnatural. Isoleucine and threonine are having two centers of chiral, two chiral centers are present. That's why they remain in the four stereoisomeric forms. D variety में आपके ग्रामिसिडीन, polymyxin, actinomycin, valinomycin, bacterial peptidoglycans, they have D amino acids present in all these proteins. Reactions of amino acid, they can be due to the carboxylic group. The reactions can be due to the amino group, because two important functional groups are attached or present in the structure of proteins of amino acid. So the reactions due to the carboxylic group, they are decarboxylation reaction, amide formation. These are the general reactions of carboxylic group you already studied in your intermediate classes. So the decarboxylation, amide formation reaction, amide formation, because aspartic acid and glutamic acid both are having in the side chain, there is a carboxylic group. So carboxylic group, one, it meets with the amino group or ammonia. If the concentration of ammonia is raised, so immediately aspartic acid picks up the ammonia, converts up into the amide group. That side chain acid, it converts to acid to amidic group. And similarly happens here. So both are very important in ammonia metabolism or scavenging of ammonia in the living beings. Especially they are very protective. These two reactions are very protective for against the ammonia toxicity in brain. The amino group reactions due to the amino group, there are transamination reaction, very important. The first very reaction of all the amino acid is transamination reaction, then oxidative deamination reaction, and formation of carbamino compound. We'll see one by one. Reactions due to the side chain, the transmethylation reaction, transfer of methyl group, and this reaction is being shown by your methionine, because in the side chain via ether linkage, the methyl group is attached. But as such, this bond is very solid. This bond is very strong. We cannot break it out. That methyl cannot be released as such. It has the methionine has to be converted up into SAM as adenosyl methionine. Then ester formation reaction of the side chain, serine and threonine in the phosphoprotein and O-glycosidic bonds in glycoprotein, because they contain hydroxy group in their side chains. So H is replaced by these via O-glycosidic linkage when it's bind with glycosidic in glycoproteins or phospho groups in phosphoproteins. Then N-glycosidic bonds in glycoproteins, reactions of amide group. So because of these reactions are basically given by asparagine and glutamine, because they have the amide group in the side chain. Then reactions of thiol groups, and thiol groups are very important groups which are present in the cysteine side chain, and they're important for stability of protein, higher level of organization via forming the interchain disulfide bonds or intra-chain disulfide bond. Look at here the decarboxylation reaction. So this is amino acid, and when this carboxylic group is lost enzymatically or non-enzymatically, it will convert up into the amine. So this is how we can convert a number of amino acid to their corresponding amine, right? Like from histidine to histamine, tryptophan to tryptamine, tyrosine to tyramine, then glutamic acid to gamma amino butyric acid. So it is decarboxylated, and it turns to very important inhibitory neurotransmitter. So these reactions happens in the biological system as well. Then formation of glutamine, I have already discussed on this. Like the glutamic acid side chain contain a extra carboxylic acid group, and this is very lavishly takes the ammonia if it's being found in the vicinity in good concentration. So glutamine synthesis is the enzyme. ATP is required, and it converts to glutamine. So it's a very important act for scavenging ammonia and transfer, and as well as transportation of ammonia. Similarly, the transamination reaction, look at here. For transamination reaction means transfer of amino group from one amino acid to one keto acid. So this reaction happens between one amino acid and one keto acid. There is a necessary condition, like two substrates have to be present. One should be the amino acid, one should be the keto acid. It can be pyruvate, it can be oxaloacetate, it can be other keto acid. So look at here, most common reaction, glutamate is having the amino group. So this amino group, alpha amino group, it now transferred to this keto group. So it turns to alanine. यहाँ पे ये जो group is being added, the ammonia is being added here. So this ketonic group is now converted into CHNH3 group. So it is become the another kind of amino acid. So keto acid converted into the amino acid, and the amino acid which has donated the amino group is now converted up into the keto group. So here there is the appearance of keto group. Very, very important reaction in the biological system. Many pathways are dependent of protein metabolism or dependent on this pathway. So glutamate converted to alpha ketoglutarate, another keto acid, and a new amino acid is being formed. So formation of non-essential amino acid is happening here. Beside alpha ketoglutarate is another keto acid, and you all know it is a very important intermediate of Krebs cycle as well. So reaction go in both the direction. So if we require the glutamate and pyruvate, so reaction go in the Biot reaction. This is how the scavenging and transferring of ammonia takes place. We can show you the another two example. One is, here is the alpha amino acid 1, that is glutamic acid. यहाँ पे there is a keto acid, can be two. It can be either pyruvic acid or it can be either oxaloacetic acid. So if it is a glutamic acid reacting with pyruvic acid, the enzyme will be alanine aminotransferase. Very, very important enzyme, transaminase enzyme is there, which is important diagnostic enzyme for liver diseases. And the reaction between these two happens and leading to the formation of alpha ketoglutarate. Glutamic will change to alpha ketoglutarate and the alanine. Pyruvic, pyruvic acid will convert up into alanine. Similarly, if reaction happens between the glutamic acid and oxaloacetic acid, it converts up into that glutamic acid will convert it up to alpha ketoglutaric acid, while this oxaloacetic acid will convert it to aspartic acid. And the reaction happens under the influence of enzyme AST or aspartyl aminotransferase, which is one of the diagnostic enzyme of heart diseases. Disulfide bridges. This is the cysteine, one cysteine, another cysteine. So the bond is created between the two cysteine facing each other. So these are the two thiol groups. Hydrogen is lost, oxidation happens, and this is the disulfide linkage appears. So formation of this kind of linkages is very, very important for sustenance of secondary, tertiary, and quaternary structure existence of proteins. Without these disulfide bonds, we cannot expect of higher level of organization of protein. These can be interchain. These are two chain, as in case of insulin. So there are both kind of interchain and intrachain disulfide linkages are available. This is in between the two different chains, A and B chain. And within the same chain, if the disulfide bond is present, so that is intrachain. Then formation of glutathione, glycine, cysteine, and glutamic acid. All these three, they joined with each other via the peptidic linkages, peptide bonds. Through peptide bond, they are linked. So gamma glutamyl cysteine glycine is the name of glutathione. Very, very important biologically active peptide. It has many important role in the detoxification reaction, maintaining the integrity of RBCs and various other functions it perform. So the reaction, the formation of this glycine by simple two peptide linkage, it is a tripeptide. Now, here I have summarized the special functions of amino acid. So, gamma aminobutyric acid is a derivative of glutamic acid and dopamine derived from tyrosine, are neurotransmitters. Histamine synthesized from histidine is a mediator of allergic responses and reactions. Thyroxine is an important thyroid hormone, which is a hormone which is derived from tyrosine. Serine forms the active center of many proteolytic enzymes. Histidine residues are important in buffering activity of proteins. So lysine group is involved in binding of coenzymes pyridoxal phosphate and biotin. So the reactions which I showed you in the transamination reaction. So beside there is a transferase or transaminase enzyme, there is a presence of coenzyme also. PLP always mediates these transamination reaction. I forgot to tell you. So PLP is the active form of pyridoxal phosphate. So it acts as a coenzyme in the transamination reactions. Ornithine and citrulline are derivative of arginine and are essential for urea cycle. Then these are the color reactions of amino acid. Ninhydrin test is given by all alpha amino acid. Biuret test is given by peptide group. Xanthoprotic test is given by benzene ring present in the phenylalanine, tyrosine, and tryptophan. So Miller's test is due to the phenol ring, which is present in the tyrosine. Aldehyde test is given by the indole ring, which is present in the tryptophan. Sakaguchi test is a characteristic of arginine and which is due to the presence of gonadium group. Sulfur test is given by thiol groups present in the cysteine. It's not given by methionine because the sulfur is very tightly bonded in via thioether linkage in methionine. So sulfur test, simple test is given by cysteine only. Nitroprusside test is due to the thiol group is always given by cysteine. Pauli's test is given by imidazole group, which is present in histidine.